Hello students, today we are going to discuss about the variations of Mendel principle. As we know very well, Mendel formulated certain laws such as the law of dominance, law of segregation, law of independent assortment. But it's not necessary that they will be applicable at all places. There are few variations where the Mendel principles are not applicable. We'll be going to discuss about that variations only, and that's why we are studying variations of the Mendel principle. But before studying the variations of the Mendel principles, you must know about the two terms, because I will be using these terms today in my video. Okay, so the first one is known as the locus. What is locus? Locus is the point on the chromosome where the gene is present, or we can say that where the alleles of genes are present. Locational point on a chromosome where an allele is located. Okay, now what is an allele? What is an allele? Any two forms of the gene, they may be similar or dissimilar, present on the same locus in two homologous chromosomes, and they are representing the same trait, is called as what? Allele. Say for I have made one homologous chromosome and I have made another homologous chromosome. Okay, now this is the locus and this is the locus. If one gene is present here and another gene is present here, so this is what? A pair of what? Allele. Okay, so any two forms of the gene may be similar or dissimilar. Here I have written dissimilar. Okay, they may be similar also. So any two forms of the gene, similar or dissimilar, present on the same locus. You can see that if capital T is present here, then on another homologous chromosome the small t is present here. So they are present on the same locus in two homologous chromosomes, and they are representing a same trait. Say for capital T and small t are representing a trait. Okay, and that is known as what? The plant height. Okay, so they are representing a trait, plant height, and the plant height is having two contrasting characters. One is known as tall and another is known as dwarf. So this is about what? The allele. And if we want to study about the variations of the Mendel principles, so we have to first study the gene interaction. Now what is gene interaction? One gene interact with the another gene and alter the phenotypic expression of the other gene, is called as what? The gene interaction. Okay, now gene interactions are basically of two types. One are known as intra-allelic gene interactions, and the another are known as non-allelic or intergenic interactions. Okay, it means we can say that they are allelic gene interactions and they are non-allelic gene interactions. Now first of all, I have a discussion here that intra-allelic gene interaction, or we can say that intragenic interaction. What are these type of interaction? Here there is interaction between two alleles of a same gene. Means I want to say in intra-allelic interactions, gene is one. Okay, and alleles are two alleles. Okay, one gene and two alleles. So interaction between two alleles of a same gene. So interaction between the two alleles of same gene is called as what? The intra-allelic or we can say that intragenic interaction. Say for I have made two homologous chromosomes. Okay, and on this locus I have written capital T and on this locus I have written small t. So gene is one, but it is having two alleles. One is known as the capital T and the another is known as small t. If the interaction is happening in between these two genes, okay, then such an interaction will be called as what? Intra-allelic gene interaction or intragenic interaction. Again I am repeating, interaction between two alleles of the same gene is called as intra-allelic gene interaction. And the best examples of the intra-allelic gene interactions are the incomplete dominance and the codominance. Now have a look here. What is intergenic interaction? What is intergenic interaction? Intergenic interaction is a non-allelic interaction. Means here the interaction will not happen between the allelic genes. The interaction will happen between two different alleles. Okay, so here two or more independent genes present on same chromosome or different chromosome interact to produce a different phenotypic expression, is called as what? Non-allelic gene interaction. Say for if this is a homologous chromosome pair and this I am making another homologous pair. Okay, now say for at this gene locus there is present a gene capital P, and at this locus what is present? A small p gene. Okay, now this capital T and small t, capital P and small p, they are not allelic genes. Okay, they are non-allelic genes. And say for if the interaction is happening in between these two genes, right, that is the interaction is happening between the capital T small t allele and the capital P small p allele, then such type of the interaction will be called as intergenic interaction. Now this interaction may also happen on the same chromosome. It's not necessary that the intergenic interaction always occur between the genes, between the non-allelic genes which are present on the different chromosome. It may happen between the genes which are present on the same chromosome. So you can see two or more independent genes present on two or more independent genes present on same chromosome, or between different chromosome. If they interact together and if they produce a different phenotypic expression, so it is called as a non-allelic gene interaction, or we can say intergenic interaction. Okay, the examples of the non-allelic interactions are many. Say for epistasis, complementary gene, supplementary gene, duplicate genes, polygenic effect, inhibitory gene effect. All these are the examples of what? Non-allelic or intergenic interactions, while the intra-allelic or intragenic interaction are having the two examples, incomplete dominance and codominance. In this video we will be discussing purely on the incomplete dominance, and it's a very important phenomenon, so we'll be concentrating on the incomplete dominance. So incomplete dominance is a sort of intra-allelic gene interaction. Okay, so see here, incomplete dominance. Okay, it is a good example of intra-allelic or intragenic interaction, right? Now incomplete dominance is also called as blending inheritance. In a couple of minutes I will be explaining you that why it is called as the blending inheritance. Okay, so first of all you must know that why incomplete dominance is called as incomplete dominance. So for this you must know that there was a scientist whose name was Carl Correns, okay, who explained the phenomenon of the incomplete dominance in a plant known as Mirabilis jalapa. This Mirabilis jalapa plant is also called as 4 o'clock plant. Okay, so Carl Correns explained the phenomenon of incomplete dominance in a plant known as the Mirabilis jalapa. He took a red flower variety and he crossed with a white flower variety. Okay, so he took two varieties of Mirabilis jalapa as parents. One was known as the red flower variety and another was known as the white flower variety. Okay, then he made the gametes. Okay, then he made the gametes. The gametes are capital R and the gametes are small r. Okay, and when we'll be crossing, we'll be getting what? F1 generation, and this F1 generation is capital R, small r. Okay, now according to the law of dominance, we know that when Mendel crossed capital T small t, then he got capital T small t in F1 generation, and he told this as tall, though it was a hybrid, but then also he called it as tall. Why? Because according to the law of dominance which was given by the Mendel suggests that in F1 generation the capital T is fully dominant over small t, means capital T allele is dominant over the small t allele, and hence we write the tall. That is, all the plants in the F1 generation will come tall. But what happened when Carl Correns crossed a red flower variety, right, of Mirabilis jalapa with a white flower variety? he got, instead of the red color, he was expecting that he will be getting the red color, because law of dominance is applicable, and according to the law of dominance, capital R is dominant over small r, so he must got what? Red color. But instead of the red color, he got pink color. Okay, now what conclusion he gave? What conclusion he gave when he got the pink color? He told that, what he told? He told that the R, capital R is incompletely, capital R is incompletely dominant over small r, that is capital R allele is incompletely dominant over small r, and because of this incomplete dominance, the capital R is not able to be fully expressed, and instead of the red color we got what? Pink color. And we know that pink color is an intermediate color of red and white color. It's an intermediate color of red and white color. Okay, so we can also say that incomplete dominance is also called as intermediate. It is also called as intermediate inheritance. Intermediate means we got the pink color, right, which is the intermediate color of red and white color. So this was an intermediate color, and that's why it is also called as intermediate inheritance, right? And the capital R was incompletely dominant over the small r. Not only this, incomplete dominance is also called as the blending inheritance. Blending means what? A mixture. Okay, pink color is a mixture of red and white color, and that's why it was also called as what? The blending inheritance. Okay, now what he did, that capital R small r, what he got pink color in F1 generation, he tried to crossed, okay, he crossed the capital R small r with capital R small r, that is he did what? Selfing, and tried to know that is there any difference in the ratio what the Mendel has calculated and what the ratio he has got here. Okay, so see here, for getting the F2 generation, what he did, he made a checkerboard. Okay, he made a checkerboard. Okay, so here capital R small r, here capital R small r. Okay, now have a cross, capital R, capital R, capital R small r, capital R small r, and small r small r. Okay, now write the names, Now this is what? Red color. Capital R small r is pink color. Okay, capital R small r is pink color, which is intermediate of red and white, and this was what? White. He got red color, pink color and white color. After getting the F2 generation, and by viewing the Punnett square, he gave the phenotypic ratio. He gave the phenotypic ratio of what? The incomplete dominance. That what phenotypic ratio we get in incomplete dominance. So here we can see what is a phenotype. Phenotype is the external makeup of an organism. We view the organism from outside. You can see that in this checkerboard, from viewing outside, we have got one red color. By viewing this thing, we can see that we have two pink colors and we have one white color. So the ratio came out to be, phenotypic ratio is 1:2:1. Now you can see that this is the deviation from the monohybrid phenotypic ratio. Okay, in monohybrid phenotypic ratio, which was actually calculated by the Mendel, there you got the phenotypic ratio 3:1. Okay, but here you can see that the phenotypic ratio of incomplete dominance is 1:2:1, means it is varying from the Mendel principles. In Mendel, we got the phenotypic ratio in a monohybrid cross 3:1. Here we got 1:2:1. And just have a look here, that genotypic ratio. What will be the genotypic ratio? Now what is genotype? Genotype is what? Genetic makeup of an organism. So we have to see the genotype. We have to see that how many capital R are there. Pure reds are there. So there is one pure red having two capital R. Now see the genotype, capital R small r. How many capital R small r that are there? So we are having two capital R small r. So these are the pink color, and we have one pure white. So this was what? The white color. Okay, so this came out to be the genotypic ratio. So we must know that in incomplete dominance, we get the same phenotypic and the genotypic ratio 1:2:1. Okay, so this was the experiment done by the Carl Correns on Mirabilis jalapa. Okay, now we have said that incomplete dominance is a type of blending inheritance, but we will say here that it is not a true type of blending. It is not a true type of blending, because you can see that in F2 generation we have got only 50% of the pink progenies, while the 25% progenies were red and 25% progenies were white. It means it is clearly indicating here that it is not a pure blending. It's not a pure blending. If it was a pure blending, then we must have got only the pink color in F2 generation, but instead of getting the entire pink color, we have got 50% of the progenies we are having the pink color, and 25% progenies are having the red color, and the rest 25% are having the white color. It means it is not a pure blending inheritance. Okay, and here you must remember this thing, that alleles are showing partial dominance. Okay, alleles are showing partial dominance. Alleles are not showing a complete dominance. The alleles are showing what? Partial dominance. Okay, so this must be also remembered, right? So this example clearly indicates that it is a variation of the Mendel principle. Okay, now coming to the next one, there are few examples. There are few also examples to explain the phenomenon of the incomplete dominance, right? Coming to the second example of incomplete dominance, and that is known as the Andalusian, called as Andalusian fowls. Okay, these Andalusian fowls are nothing, they are the poultry fowls, right? They are the poultry fowls. Fowls are nothing, they are the birds which are kept in the poultry. Okay, so we will be taking an example of Andalusian fowl to explain the phenomenon of the incomplete dominance. Okay, so the Andalusian fowls are having two varieties. Okay, one is known as capital B, and another is the small b. This is for the black color. This is for the black color fowl, and this is the parent for the white color. Okay, so these are the two types of the poultry fowls. Okay, while crossing, what we see that these are parents. These are parents. Now we can make the gametes here, capital B and small b. Okay, now when we cross, then what we get? Capital B, small b, and this is F1 generation. If Mendel was correct, then we must have the color in F1 generation, we must have got the black color, but instead of the black color we got which color? That is known as the blue color. It was not purely blue color, but the feathers of the fowls were having the stripes of the black and the white in alternate fashion, so it was seeming to be blue color. Okay, so F1 generation we got blue color. If Mendel was correct, we must have got black color, but instead we got blue color. It is clearly indicating that capital B allele is incompletely dominant over the small b allele. If the capital B was completely dominant, then the color must have produced to be the black color, but here the black color is not produced because the capital B is incompletely dominant over the small b. So therefore an intermediate color of black and white was produced, and this intermediate color was known as what? The blue. Okay, now see here, if we cross the parents, okay, then what type of the progenies we get? See here, if we cross the parents, capital B crossed with small b, so capital B, small b, capital B, small b, capital B, small b, capital B, small b. So we can see here that all the progenies, all progeny we get what? Blue color. All progeny we get what? Blue color. Now if F1 generation is crossed, okay, if F1 generation is crossed with the same genotype, capital B, small b, then see what type of the progenies we get in F2 generation. So we will be making a Punnett square here. Okay, see here, capital B, small b, and capital B, small b. Okay, here the parents were crossed. Okay, here we have self it. Okay, we have interbreed the capital B small b with the capital B small b. So you can see here capital B, capital B, capital B small b, capital B small b, and small b small b. Okay, this was a black color progeny, this was a blue color progeny, this was a blue color progeny, and this was a white color progeny. Okay, here also you can see that the phenotypic as well as the genotypic ratio, phenotypic as well as the genotypic ratio is same, just as what we got in the mirabilis chalapa. Okay, so the phenotypic as well as the genotypic ratio is same, that is 1:2:1. Again we can say that it is a deviation from the phenotypic monohybrid cross ratio. There we got 3:1, but here we got 1:2:1. That is phenotypic ratio we got 1:2:1, and here the phenotypic and the genotypic ratio are same. So we can say that Andalusian fowls are also presenting a good example of gene interaction, intra-allelic gene interaction. Okay, now see what the poultry members did, what they did. Okay, what they did, as the blue color bird or the blue color fowl was meant for delicacy. Okay, the blue fowl was meant for delicacy. Okay, delicacy means what? It was a more better breed, it was a more better breed. Okay, the black color fowls and the white color fowls were not having so much of the market value, but the blue color fowls were meant for delicacy and they were having a good market value. So what the owners of the poultry farm did, what they did, they killed the black color and the white color fowls. They killed the black color and the white color fowls. What they were thinking, because they were not the biology students, so what they have thought, what they were thinking, that if we will be killing the black and the white color, and if we will be crossing only the blue progenies, if we will be crossing the, or we will be doing the interbreeding between the blue progenies, then we will be getting the blue color progenies only. But they were wrong, because they were not the biology student. You can see here that when a blue fowl is crossed with a blue fowl, that is when capital B small b and capital B small b are crossed, then we are not getting at all the blue fowls. You can see that we have got only 50% blue fowls, we have got 50% blue fowls. Okay, and we have got 25% black and the 25% white. So they were expecting that they will be getting all the blue fowls, but because we know very well that here also there is no true blending, there is no true blending. Okay, that there is no true blending. If there may have true blending, then we may have got what, the 50 that is, instead of the 50% blue fowls, we have got 100% what, blue fowls, but we have got 50% blue fowls only. Okay, and what they did, they killed the black color and the white color fowls. Why they killed? So that they may not mate with each another, right? They were thinking that by killing the black and the white color, okay, only the blue progenies will be crossed in the poultry farms and they will be getting the blue color. But they were wrong at their side. Whenever a blue fowl is crossed with a blue fowl, only 50% of the fowls will be generated, will be of the blue color. So Andalusian fowl is also a good example of incomplete dominance. Okay, now the third more example, one more example is available to explain the phenomenon of the incomplete dominance, and the third example is of the snapdragon, right? The snapdragon is also called as dog flower. It is also called as dog flower, or in botany it is also called as Anthyrhinum. It is also called as Anthyrhinum majus. Okay, so when a broad leaf variety is crossed with a short leaf variety, right? So these are what, the parents, these are the parents to make the gametes right? The gametes will be capital B and the gamete will be small b. Okay, so capital B, small b. Now according to Mendel, right, if we trust on the Mendel, the F1 progeny must be broadleaf variety, but it was not a broadleaf variety. We got a intermediate type. We got a intermediate type of leaf, which was not so much broad, which was not so much short. This intermediate type of the F1 progeny again indicate that the allele for the broad leaf is not fully dominant over the allele of the short leaf. Okay, and that's why the capital B is not fully dominant over the small b, and instead of the broad leaf we got a intermediate leaf type in the F1 progeny. And when it will be interbreeding or selfing the capital B, small b, right, then in F2 generation we'll be getting again the ratio 1:2:1. So by the above discussion we can say that the incomplete dominance is a variation of the Mendel principle. So we have quoted 3 examples of incomplete dominance. One was known as the Mirabilis jalapa, okay, the second was known as the Andalusian fowl, and the third one was known as the snapdragon or the dog flower. Okay, so we have purely concentrated this video on the incomplete dominance. We'll be discussing few more gene interactions like complementary gene effect, supplementary gene effect, epistasis, inhibitory gene effect, codominance, multiple allelism in the next videos. Keep watching. Thanks a lot.